Hello everyone, my name is Phillippe. I'm from University of Brasília. My project is entitled Proteomic and Functional Analysis of IL-8 activated neutrophils. So we can know that neutrophils stimulated by IL-8 be involved in different pathologies, such as acute respiratory distress syndrome. So we analyzed the molecular mechanisms that follow IL-8 stimulation in neutrophil stroke, a comparative proteomic analysis associated with functional tests. This analysis identified 39 proteins that showed significant difference abundance, P less than 0.05, between stimulated and resting cells. We analyzed the proteins by Panther-GO and Chin-GO. The results obtained from this analysis showed use the profile of proteins regarding molecular function, biological process, and cellular component. Most regulated proteins are linked to structural and catalytic activities in agreement. Most proteins are involved in processes such as vesicle-mediated transport. The majority of proteins are present in the cytosol cell membrane and chromatin. In addition to this, analysis Chin-GO also revealed functionalities such as neutrophil degranulation and response to cellular stress. IL-8 affecting neutrophils is controversial with different data about whether IL-8 is able to fully activate of the NADPH oxidase complex and consequently induce ROS production. Based on these studies, we evaluated the release of ROS using nitroglutathione. The results show a significant difference, P less than 0.001, in the presence of formazan deposits between control and IL-8 stimulated cells. The crystals are more intense and mostly localized close to the cell membrane of neutrophils stimulated by IL-8, while few control cells show a low intensity activation close to the cell nucleus. Spectrometry analysis confirmed the results observed by microscope between the groups, P less than 0.05. Moreover, the proteomics results that are related to cellular stress pathways showed upregulate proteins SERPIN1, ARM1, HP, H1M, HRG, act in the inhibition of the NADPH oxidase pathways, preventing the release of ROS. Therefore, although we corroborate the activation of NOx in IL-8 stimulated neutrophils, we hypothesized that this process is strictly modulated, and such a modulation demands further studies. I would like to thank all the collaborators who participated directly and indirectly in the work, as well as the funding agencies.